This is a production of Cornell University.
Thanks, Anna. Today I'm going to be talking about my ongoing project examining the regulation of carotenoid content and its interactions with carbohydrate metabolism in cassava. So Emily did a nice job setting us up for this one. Thanks, Emily. So cassava is known as a food security crop because it's a really important source of carbohydrates and calories for many tropical areas of the world. It is rich in starch, but not many other micronutrients. So as a result, a lot of populations that rely heavily on cassava-based diets tend to have micronutrient deficiencies. And one of those in sub-Saharan Africa in particular, provitamin A deficiency is affecting over half of children under 5. So it's a really common deficiency among people who eat cassava. But there are varieties of cassava that naturally accumulate some provitamin A content. They have a yellow pigment, and that not only boosts the nutritional quality, but also improves the post-harvest storage of cassava. So cassava breeders have been working on improving yellow cassava to increase the beta carotene content, which is cleaved in half to produce vitamin A inside the human body. So it's considered provitamin A biofortified. And through recurrent selection, breeders have been doing a pretty good job at increasing carotenoid content, but it's been difficult to combine high carotenoid content with other favorable traits that are needed for consumer acceptance. In particular, carotenoids are negatively correlated with dry matter content. And so this makes it challenging to combine both high carotenoids and high dry matter content. Which dry matter content is essential for consumer acceptance of new varieties. It influences the dry yield and also the textural properties. So my project comes in to see how can we improve both carotenoid and dry matter contents simultaneously in cassava breeding. So for this, it's important to know what is the genetic relationship between carotenoids and starch, which are negatively correlated. So this could be driven by linkage, genetic linkage, or pleiotropy. So in genetic linkage, the hypothesis is that there are two separate loci close together on the chromosome, one that influences carotenoids and one that influences starch. And there are such loci that have been identified in cassava. One is the phytoene synthase 2 allele, which Emily introduced. Phytoene synthase 2 is the rate-limiting step in carotenoid biosynthesis. Nearby this allele, there's also 2 candidate genes for carotenoid, for carbohydrate synthesis, including sucrose synthase. And so it's thought that there's a PSY2 allele that increases carotenoids and a linked sucrose synthase allele that decreases starch. There's also evidence that phytoene synthase expression itself could also affect carbohydrate metabolism. So in cassava and potato, when PSY is overexpressed, there's a dramatic increase in carbohydrates and a concurrent decrease in starch, as well as a lot of other broad metabolic shifts, changes in carbohydrate metabolism, fatty acid metabolism, and broad changes in gene expression associated with those metabolite pathways. And carbohydrate metabolism is also affected in several other crops that have been engineered to produce higher carotenoids, including tomato, citrus, and maize and rice. So I think it's important to distinguish between these two scenarios because if we are dealing with genetic linkage, you could theoretically break the linkage by continued breeding and continued recombination, potentially aided by markers. If it is an inherent pleiotropic relationship that affects the metabolism, then you may need a metabolic engineering approach to increase both simultaneously or to focus on other QTL besides the PSY2 allele. So just briefly to mention some of the hypotheses about how PSY2 could be pleiotropically affecting carbohydrate metabolism. So one of the main hypotheses of the authors who transgenically modified the yellow cassava was that this UGPase, UDP glucose pyrophosphorylase, the expression of that enzyme is being affected by carotenoid accumulation through an unknown regulatory mechanism. There's also other hypotheses involving the co-location of carotenoids and starch in the same plastids. So carotenoids and starch are both synthesized in amyloplasts, and precursors for both those processes need to be transported across the membrane. So there could be local competition for glucose or ATP within the amyloplast, or downstream products of beta carotene like abscisic acid could be having the broader effects on metabolism. On the other side, if it's true that genetic linkage is mainly driving the correlation, then we want to understand how that locus is affecting the trait and also what other loci are affecting the trait so that we can improve carotenoid content without decreasing dry matter. So the distribution of carotenoid content as estimated here by a chromometer B value, it's bimodal. So on the left, there tends, the white cassava is generally considered to be less than 20. And the yellow cassava is generally considered to have higher carotenoid content. And we know that that difference between white and yellow is driven by the PSY2 allele. All of the yellow cassava have at least one copy of that allele, and it's additive. There's also some quantitative variation in carotenoid content within that yellow cassava group, and that's what I'm interested in looking at. So there's a range and gradient from cream to yellow, and within that, there's also some interesting spatial variation. So we have this fun phenotype called the egg yolk phenotype, where the center tissue layer of the root is yellow and or darker than the outside. So it looks like an egg yolk. And that's one example of spatial variation in carotenoids that we know isn't caused by the PSY2 allele because it's happening, different accumulation is happening within the same genotype. So I want to use this system of the inner and outer tissue layers to test some predictions. The first is that the tissue-specific carotenoid enrichment is transcriptionally regulated. So I want to see what genes are upregulated or downregulated in the center layer that's enriched with carotenoids compared to the outside. And then I'm also predicting that if there is an inherent metabolic trade-off between carotenoids and carbohydrates, then I would expect that to persist across spatial and environmental variation as well as genetic variation. So if there's an inherent trade-off where the plant only has so much resources it can transport into the amyloplasts, if it's focusing more on carotenoid biosynthesis in the internal tissue layer, I expect that internal tissue layer to have lower dry matter content. So to test some of these predictions, I set up a greenhouse experiment. And the main objectives of this is to understand the regulation of carotenoids beyond the PSY2 locus, looking at the quantitative variation, and also to test hypothesized interactions between carotenoids and carbohydrates. So for this experiment, I have in the greenhouse a set of 54 accessions. 5 of them come from Africa, 5 of them come from Colombia, and then the rest are hybrid crosses between them. And I've grow these in greenhouses in pots, measure the carotenoid composition, measure starch and sugars, and I'm working on extracting RNA for 3' RNA-seq. I'm repeating that trial with a subset for a second year, and I'm going to harvest at different time points and then do an expression analysis I'll talk more about. So for the metabolite quantification, for these cassava samples, I separated them into inner and outer tissue layers using this makeshift cork borer, and then lyophilized the tissue, and so measured dry matter percentage by the dry weight divided by the fresh weight. I extracted and measured carotenoids in Ted Tannhauser's lab at the USDA with the help of Tara Fish. And so we measured those with UPLC UV-Vis. And I'm working on measuring starch and sugar with colorimetric assays. So from this preliminary trial in 2021, we first just grew out the seeds so that we could get them all in a clonally propagated state and did an initial characterization of the germplasm. And we did see a negative correlation here between total carotenoids and dry matter content. And so that was good. We also saw that there was variation in carotenoid composition. So we noticed that the African cassava lines tended to accumulate more of these precursors, the phytofluene, and the Latin American varieties tended to accumulate more of the violazanthin and lutein. And most of the variance is explained by variation in trans-beta carotene, which is the major carotenoid compound in cassava. So then we propagated this trial and replicated it in 2022. And I was surprised to see that there was basically no relationship between carotenoids and dry matter in this trial. If anything, it was slightly positive. And the heritability for carotenoids was relatively high, 0.8. It was pretty low for dry matter, 0.175. So there's a lot of things that could have been different between this trial and the last preliminary trial. There are different greenhouses, different pots, different replicates. And of course, we separated these by tissue layers where we didn't separate the other one. So there is some environmental variation going on here for sure between the first and the preliminary trial. But I was interested to see that in this case, there is no apparent correlation. Looking at the tissue layer, I saw that the relationship between carotenoids and dry matter differs depending on which tissue layer you're looking at. So in the inner layer, it is negatively correlated, but in the outer layer, it's actually positively correlated. So perhaps the developmental stage of the root is really important. And if you see that overall correlation, because it might depend on the ratios of the inner and outer tissue layers and how big those are and how much you sample. So moving forward, I'm going to sample at different time points so I can try and capture the growth of the root and how that changes over time. I'm also working on doing RNA-seq from the inner and outer tissue layers. Between inner and outer tissue layers, I want to look for differentially expressed genes. So I'm looking for genes that are expressed in the enriched inner tissue layer. That might give us a clue about how carotenoids can be increased in cassava and what genes are important. Second, I want to look for trait-associated genes. So those are transcripts that are associated with quantitative variation in carotenoid composition as well as amount. So we want to maximize beta carotene since that has the highest provitamin A activity. And then I want to do a network analysis to look at the co-expressed gene modules and identify if there are carotenoid and carbohydrate metabolism enzymes in the same modules, which would suggest a pleiotropic relationship. So right now we're growing the second trial. It's in the greenhouse where we use a subset of 20 genotypes and they have been genotyped. We're harvesting them this summer at 4 different time points, and they're going to replicate the analysis from trial 1 with the metabolites and RNA-seq. And moving forward after harvesting that trial, I'm also looking at doing a multivariate GWAS in the breeding population at the International Institute of Tropical Agriculture so that we can look across the genome and across more genetic diversity at what other loci might be affecting carotenoids and dry matter either jointly or independently besides the PSY2 locus. And then finally, I want to synthesize the results from that GWAS with this expression analysis to develop some suggesting breeding strategies for dealing with this trade-off in cassava. So that might mean developing markers to help break the recombination, to break the linkage and screen for recombination. It might mean identifying additional QTL besides the PSY2 locus that don't have effect on dry matter, or perhaps identifying metabolic engineering targets. And with that, I'd like to thank my committee, the Tannhauser lab, the Guterman staff. I've worked in several labs to do this wet lab work, the Soros lab, the Giovannoni lab, and the Gore lab. And I'd like to thank our collaborators at NextGen Cassava and IATA who have provided the materials. And finally, I need to mention Jiho Lee, who's pictured, helping me harvest the cassava. He's an undergraduate student mentee who's been helping me with this project last summer. Thank you. Travis?
Yeah, I think you mentioned this a little bit, Seren, but do you think it caused an effect that you're using this constant size sort of, right? You're separating core and outer core. Do you think if that has an effect, maybe separating the things that are certainly outer and certainly inner in this little space.
Yeah, so the question was being certain about separating the inner and outer tissue layers. The way I've been doing this, it's definitely not exact. The way I've been separating the layers, sometimes you can see it visually, like if the inner color is different, you can see where that line is. And I have different size diameters of those pipes and try and match the closest one, but it's not perfect. And if I can't see the inner tissue layer, I guess based on the proportion and other similarly sized ones, what size I use for them. So it's not capturing like the exact tissue layer, but I think it's probably separating the general tissues enough to, if there's a different relationship, to be able to capture that. Yeah.
Yes, no. Inner outer, you could look at or variable tolerate, which might give you stronger correlation, or which might give you a stronger between the two tissues. Is that something you could look at, or are you really looking at the spatial?
Yeah, I'm mostly interested in the spatial difference because it's that the egg yolk phenotype has been something that we know is not driven by PSY2, but we don't know what causes it. There are other spatial variants, like if you go long ways in the root, there's some variants from like the proximal to the distal end. But I just decided to focus on this one just because I can't do too many segments or I'll have a million samples. Emily? Yeah, it's in the coding sequence and it changes the catalytic capacity of the PSY2 enzyme itself. Matthew?
Have you looked at any genes in the plastid genome that might be affecting the interaction between, or do you have any genes that might be controlling the transition from amyloplastic to chromoplast?
Yeah, the question is if I'm looking at any plastid genes. I'm definitely interested in looking at the expression of OR. I've seen in my, I've done some microscopy on the roots, and I haven't seen clear evidence of chromoplast, but I have seen some evidence of chromoplast. So there's been I've observed some crystals forming, some carotenoid crystals, which is suggestive of at least amylochromoplast, but I guess the line is quite blurry. So I'm interested to see if I can learn more about that, looking at expression of OR. And if you have any other plastid associated genes you know of, I'd be happy to hear about them. Mike?
I feel pressure now since I'm sitting somehow at the Cassava table.
Is there actually a large variance for PSY2 and Cassava germplasm, or is there essentially only one haplotype for PSY2 in Cassava?
Yeah, so the question is if there's a lot of variance, allelic diversity with PSY2, or if it's just that one haplotype. I'm only aware of that one haplotype in all of the yellow cassava sequences I've looked at. I've just looked at the coding region, and it is that same SNP, but there are possibly other SNPs and other accessions I haven't looked at, or expression differences that I'm not aware of. Yes.
Do all carotenoid species
Do all
all carotenoid species negatively correlate with the carbohydrates? Is there any particular carotenoid?
Yeah, so the individual carotenoids tend to all follow the same relationship as total carotenoids, which is mostly beta carotene. I tried looking at all the different components to see, okay, maybe phytoene is negatively correlated or something, but they're all pretty much following the total carotenoids. Is that answering your question? Yeah, so in cassava, starch is the main component of dry matter, but there are also other components like fiber. And so that's something that could be affecting I'm using dry matter here as a proxy. So I'm working on measuring directly the starch content so I can try and distinguish between starch or sugar specifically compared to other components of dry matter. Okay, thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.